Hello, everyone, and welcome to today's session. And today we'll be discussing the NCERT exercise for the chapter Principle of Inheritance and Variation. Okay, so this is the part 2 of the lecture. So, what are the objectives of today's session? We will explore and understand the fundamentals concept of chapter 4, which is Principle of Inheritance and Variation, by working through NCERT exercises. Okay. So these questions are based on some concepts. We will understand these concepts by discussing the NCERT exercises. So we have discussed from question number 1 to 8 in the previous session. Today we'll be discussing question number 9 to 16 in this session today. Okay, so let's begin with this question now. This is question number 9 of the exercise. So question is, briefly mention the contribution of T.H. Morgan in genetics. So T.H. Morgan, he was experimenting on Drosophila. So he did experiments on Drosophila melanogaster, which is a model organism. Now, what was his contribution in genetics? Okay, for more information, you can read NCERT section 4.3.3. So what will be the answer for this question? So let's have a look at the answer now. So contribution of T.H. Morgan in genetics were, so Morgan work was based on the fruit flies. What is the scientific name of fruit flies? Drosophila melanogaster. So a lot of experiments are carried out on Drosophila melanogaster. Why? What is the reason for that? Because Drosophila melanogaster, it has a short lifespan. It produces many progeny. Okay. The sexes are easily distinguishable. So you can distinguish male from the female. Okay, so these are some of the reasons why Drosophila melanogaster is used to carry out research work. Then this Drosophila melanogaster, this can be easily cultivated on a very simple medium. Okay, cultivation of Drosophila doesn't require much space as well. Crosses can be made easily. Okay, so Morgan, he was studying the genes present on the X chromosome. So he was studying the sex-linked inheritance. And he formulated the chromosomal theory of the linkage. So Morgan, he discovered linkage and recombination as well. So both of them, linkage and recombination, was described by Morgan by his experiments on fruit flies. So he conducted dihybrid crosses, which was similar to the dihybrid crosses made by Mendel in the pea plant. Okay. Now, he defined linkage. So how he defined linkage? As the coexistence of two or more genes in the same chromosome. So these linked genes, he said these linked genes, they are present on the same chromosomes. So that's why it's written coexistence of genes. Coexistence of two genes or more than two genes. And these two genes, they are present very close to each other. The distance between the two genes is less. Okay, that's why the recombination frequency is very less. Okay, more the distance between the genes, recombination frequency will be more. Percentage recombination will be more. If the distance between two genes is less, then the percentage recombination will be less. Now he performed dihybrid crosses in Drosophila. Okay, similar to dihybrid crosses that Mendel made in pea plant. So what is a dihybrid cross? A cross which involves two traits. And these two traits, they are controlled by two genes. So in dihybrid crosses, the organisms which are being crossed, they differ in two traits or two genes. And in dihabet crosses, we study the inheritance of two genes. Clear? So, he showed that the linked genes are inherited together, and these linked genes are located on the X chromosome. So, Morgan was studying the genes present on the X chromosome. So, he was studying sex-linked inheritance. And these genes, they were inherited together. So, how he concluded that the genes were linked? Because in its experiment, Morgan was getting more parental combinations as compared to the recombinants. And the phenotypic ratio in the F2 generation, it deviated from the normal 9:3:3:1 ratio. So what Mendel said, if the genes are assorting independently, okay, if they are not linked together, then in the F2 generation, 9:3:3:1 is the phenotypic ratio. But in the case of linked genes, you don't get this ratio of 9:3:3:1. So if there is a deviation from this ratio, you can say that genes are linked and you will get more parental combinations. Okay. Now these two genes can be tightly linked or they can be loosely linked. So genes, if they are tightly linked, they show very low recombination. And if the genes are loosely linked, then genes show higher recombination. Okay. So here you can see in this figure, this is showing you inheritance pattern of the linked genes. So these are very tightly linked genes here. One is female, another one is the male. Male consists of the XY chromosome, female consists of XX chromosome. You can see this Y and W genes. The distance between these two genes is very less. And you are getting more parental combinations. Parental combination is 98.7%. Recombinants are 1.3%. So these genes are very tightly linked. Okay. In another case, the genes can be loosely linked. So here in loosely linked genes, you will certainly get more number of parental combinations. Okay. But they will be not like 98.7%. They'll be around like 62%, as we have discussed in the theory. Okay. The next question, what is pedigree analysis? And suggest how such analysis can be useful. So this question is based on pedigree analysis. Okay. So by pedigree analysis, you can study inheritance of traits, you can study inheritance of abnormalities, diseases, genetic disorders. Okay. So how this pedigree analysis can be useful? So what will be the answer for this question? Okay, let's have a look at the answer now. Now, what is pedigree analysis? Analysis of traits in several of generations of a family. So whenever you are analyzing the traits or inheritance of a particular trait, inheritance of a particular gene, inheritance of a particular disorder in multiple generations or over several generations in a family, this is known as pedigree analysis. So you get a family tree here. Okay. So what you have to remember in pedigree analysis, you are analyzing the traits or the genes or the genetic disorder over several generations of a family. Now, it is based on the fact that certain characteristic features are heritable in the family. Okay. Some of the features, they are inherited, means they are passed on from the parents to the offerings. So usually the DNA is passed on from the parents. It is the DNA which is passed on from the parents to the offerings. Okay. Or you can say genes are passed on from the parents to the offerings. What are genes? They are the segment of DNA. Okay. So this pedigree analysis is based on heritable material, which is the genes or DNA or chromosomes. Genes are present on the chromosomes. For example, eye color inheritance. If you want to study the eye color inheritance, okay, in this, the geneticists, they can tell what is the probability that the blue color eye will be, you can say, the offering will inherit blue color eye or brown color eyes or black color eyes. Then skin color, hair form, and hair color, and other facial characteristics. Apart from this, you can also trace the inheritance of genetic disorders and abnormalities. What is the significance of pedigree analysis? What is the importance of pedigree analysis? It allows to study the inheritance pattern of specific traits, abnormality, or the disease in a family. Here, so you can study the inheritance pattern of a specific trait. What do you mean by inheritance pattern of a specific trait or abnormality or disease in a family? Means you can you'll be able to understand how that gene is being inherited from parent to the offerings. Okay. How the genes means, is the pattern of inheritance recessive? Okay. Is that gene following dominant pattern? So we talked about the inheritance patterns, okay, such as autosomal dominant inheritance pattern, autosomal recessive inheritance pattern, then sex-linked inheritance pattern. So pedigree analysis gives you this information, how that character, how that trait, or how that gene, or how the disorder is being inherited. What is the pattern? Clear? Whether it is being inherited from mother to the sons, okay, or from father to the daughters. Now, in this analysis, inheritance of a trait is represented as a tree called the family tree. So you get the family tree here. Family tree means you are analyzing a particular trait over several generations. Your grandparents, parents, and then offerings, and then their offerings. So you get a family tree here. Now, genetic counselors use pedigree chart for analysis and prediction of various traits and diseases in a family. So it can be used for genetic counseling. So these genetic counselors, they can use this pedigree chart and they can find out, they can predict that what will be the probability of occurrence of a particular disease, or what is the probability of occurrence of a particular trait in the offerings. Is there 25% probability, 50% probability, 0% probability, or 100% probability? So the genetic counselor can predict it. And then required measures can be taken. It is useful in preventing hemophilia. So these are all genetic disorders. X-linked recessive genetic disorder, hemophilia, sickle cell anemia, autosomal recessive disorder, and other genetic disorders in the future generations. Because these genetic disorders, they are inherited from one generation to another generation, from parents to the offerings. Now, next question. How is sex determined in human beings? Okay, basically, or you can say, how is gender of the child determined in the human beings? So what will be the answer to this question? And for more information, you can read the NCERT section 4.6.1. Okay. So let's have a look at the answer for this question now. So how is sex determined in human beings? Sex determination in humans, you know which sex determination system humans follow? XX and XY sex determination system. Here the females, they are XX, they have a pair of X chromosome, and males, they are XY. They have one X chromosome and one Y chromosomes. So females, as they are having two X chromosomes, they are homogametic. Means they are producing only one kind of gametes. All the eggs will contain only X chromosome. Whereas males are heterogametic. Means males are producing two kinds of gametes. Clear? As males have X and Y, so half of these sperms will carry X chromosome, half of the sperm will carry Y chromosome. And it is the male who determines the sex of the child, because male are producing two kind of sperms. Okay. So in human beings, you can see male heterogamety. Means males are producing two kinds of gametes. In humans, males have X and Y chromosome. They produce two types of gametes. Half of the sperm will have X chromosome, half of the sperm will have Y chromosome. So this is following which law? The question can come from here. Okay. So during gamete production, what is happening? There is a 50% chance that half of the sperm will carry X chromosome and half of the sperm will carry Y chromosome. So this is following which law of Mendel? Which Mendelian law this is following? Okay, so basically this is following Mendel's law of segregation, which says that during gamete formation, half of the gamete will receive one allele, half of the gamete will receive another allele. Okay, clear? So these are the concepts on which these questions are based on. You should know all of these concepts. Now, the human females, they have a pair of X chromosomes. They produce only one type of gametes, means all the eggs will contain only the X chromosome. Now, the sex of the baby, the gender of the baby is determined by the type of male gamete that fuses with the female gamete. Means the type of sperm that fuses with the egg. Okay. If the sperm carrying X chromosome, this fertilizes the egg, then the zygote will be formed and then zygote will contain X and X. And this will develop into female child. If the sperm carrying Y chromosome fuses with the egg, then the zygote will contain X and Y and this will develop into male child. Clear? So if the fertilizing sperm contain the X chromosome, that the baby produced will be a girl. If the fertilizing sperm carries the Y chromosome, then the baby produced will be a boy. Now, hence, it is a matter of chance that determines the sex of the baby. So it is a matter of chance, you can see. There is a 50% chance that a boy will be born or a girl will be born. And usually females are blamed when the mother gives birth to the female or the girl child, then females are blamed. But it is actually the male who determines the sex of the child. So this myth should be eradicated. Education should be given regarding these issues, especially in the rural areas. Yeah. Now, there is an equal probability of fertilizing sperm with an X or the Y chromosome. So there's an equal probability. There is a 50% probability that the sperm carrying X chromosome will fuse with the egg and the sperm carrying Y chromosome will fuse with the egg. Clear? Thus, it is the genetic makeup of the sperm that determines the sex of the baby. Clear? It is the it is the sperms of the male that determine the sex of the baby, type of sperm. So in this image, you can see this is sex determination system in humans. Father and mother in sexual reproduction, two parents are involved. There is a biparental condition. Both father and mother will produce the gametes. Male gamete are sperm, female gametes are eggs. Males, they have XY chromosome, female, they have XX chromosome. Now males will produce two kind of gametes, female produce only one kind of gamete. Now you can see these are all the probabilities. Clear? And there is an equal chance that the offerings can be son or a daughter, male or a female. Clear? So if this X chromosome, if the sperm carrying X chromosome, this fuses with the egg, then it will form the XX condition in the zygote. This will develop into female. If the Y chromosome, this fuses with the egg, then a son will be born. Clear? And you can see here males. Remember, this is very important. Male, they have XY. Male give Y chromosome to the sons only. Okay, and X chromosome to the daughters. The question can come from here. So if there is a mutation on the X chromosome of the male, okay, if there is a mutation on X chromosome of the male, then how many sons will be affected? The answer will be 0, because male will only transfer this X chromosome having the mutated gene to the daughter, not to the sons. Male will always give Y chromosome to the sons, not the X chromosome. X chromosome it will give to the daughters. Okay, so daughter can become carriers. Okay, mostly daughters are carriers. The next question, a child has blood group O. If the father has blood group A and the mother has blood group B, you have to work out the genotypes of the parents and the possible genotype of the other offerings. Clear? So they have given, what is the information given? First you have to write. Okay. Information given is that the child is having, one of the child is having the blood group O. You have to calculate the genotype of the other children's. And you have to also calculate the genotype of the parents, means father and the mother. And they are saying father is having a blood group A and mother is having blood group B. So what will be the answer for this question? So let's have a look at the answer now. Okay. Now this question is related to ABO blood group system. Now the blood group characteristic in human is controlled by 3 set of alleles. And out of these 3 alleles, 2 alleles are codominant, IA and IB. These 2 alleles, they are codominant. Okay, usually a gene have 2 alleles, but here a single gene is having 3 alleles. This is an example of multiple allelism also. Okay. So these are the 3 alleles, IA, IB and I. These are the 3 alleles. So that's why this is example of multiple allele as well. When a gene have more than 2 alleles. Okay. The gene is I gene. Clear? This I gene has 3 alleles, IA, IB and I. Clear? Now the person having blood group A. In this person, the RBCs, the plasma membrane of these RBCs will carry a different kind of sugar. In the person having blood group B, the RBCs in this person or the plasma membrane of the RBCs of this person will have different kind of sugar. And the person having blood group I, in this person, the plasma membrane of the RBCs doesn't produce any sugar. And in the case of blood group AB, where both alleles are present, IA and IB, then in this case, the person, the RBCs consist of both the sugars produced by A allele and B allele. Okay, and IA and IB, they show codominance. IA is dominant over small I and IB is dominant over small I. Okay. And for the blood group O, both these small I alleles will be present together because this is the recessive allele. I is dominant over I and IB is also dominant over small I. So the alleles IA and IB are equally dominant. Means this show co-dominant. Both of, okay, in the heterozygous condition, both the IA and IB, both of them are expressed. That's why the blood group will be AB. So RBCs of this person will carry both the sugars on its plasma membrane. Whereas allele I, this is recessive to the other alleles. And recessive condition is only expressed in the homozygous condition. So small I, this will be the blood group O. Now, according to alleles codominancy rule, the individuals with the genotype IA and IA and small I have blood group A. Okay, so this is basically the law of dominance now. Okay, codominance is between IA and IB. So the individuals with genotype IA and IA and small I will have blood group A because IA is dominant over small I. While the individuals with genotype IB, they will have blood group B, and the individuals having IB and I, this will also have blood group B because IB is dominant over small I. Okay, so you can see this ABO blood group system. This is following law of dominance also. Okay, it is following law of codominance also in the case of IA and IB. And it is following multiple allelism also. So you can get a question. This ABO blood group system follows which laws? Law of dominance, codominance, and multiple allelism. Okay, or they can say which of the following are related to ABO blood group system. Now, while those with blood group O have the genotype small I. Now, according to the question, the child is having O blood group. Means the child will have genotype small I and small I. Only this case, you will get the blood group O. Okay. Now that means that small I allele must have come from each parent. So you know the offerings, they inherit one allele from each parent. Okay, one allele they inherit from father, another allele they inherit from the mother. Okay, clear? So if the offerings, if the child has blood group O, then it has two small I alleles. It is homozygous for small I. So the parents, both of the parent must have at least one small I in their genotype. And the question they are saying the father is having blood group A. The father should be IA small I. And the mother is having blood group B. So mother should be IB and small I. Should be the genotype of the mother. So this indicates that each parent must have small I allele. Both the parents should have at least one small I allele. So you can see here, we can see this image and we can understand this concept. These are the parents, father and the mother. Father is having blood group B, mother is having blood group A. Okay, or you can write opposite of this also. Father can have A blood group or mother can have B blood group. Clear? Now the genotype should be IB, I and IA small I. So gametes, these will be the gametes produced. So in case of IBI, half of the gamete can contain IB, half of the gamete will contain small I. In case of IA and small I, half of the gamete will contain IA, half of the gamete will contain small I. Now, you can make all the different combinations. In the F1 generation, in the first generation, you will get this child. This child is having small I. So this will be having O blood group. Okay. Now, you have to calculate the genotype of the other offerings as well. So other offerings can have the genotype what? They can have IA, this AB, B and A. So therefore, genotype of father is IA small I. Okay, clear? And genotype of mother will be IB and small I. Clear? You can just reverse it. You will get the same answer if you even reverse this. Okay, you will get the same answer here. And genotype of other child will be IA. Okay, AB. AB will contain both the alleles, IA and IB. Okay, one of the child can have blood group B, having the genotype IB small I, and one of the child can also have blood group A, having a genotype IA and small I. And one of the child will be having O blood group because it has inherited one small I allele from father, one small I allele from the mother. Clear? So this is how you calculate the genotypes of the offerings as well as genotype of the parents. This is very important question here. The next question, you have to explain the following terms with help of the examples. You have to explain what is codominance and you have to explain what is incomplete dominance. So both of them are deviations from the Mendel's law of inheritance. Mendel talked about the law of dominance, that one of the gene masks the expression of the another allele. Okay, so these both are deviations. So what will be the answer for this question now? Okay, so let's have a look at the answer. Codominance. Let's talk about codominance first. It is a phenomena in which both the alleles of a contrasting character are expressed in the heterozygous condition. Okay. Both the alleles are expressed in the heterozygous condition. So you have to write this heterozygous condition as well. Okay, example, example, this is a very classical example of AB blood group. Here both the alleles are codominant. Both the alleles are expressed in the heterozygous condition. Yeah. Now here in codominance, both the alleles of a gene are equally dominant. That's why both the alleles are expressed in the F1. Example is ABO blood group in the humans. Now the blood group character is controlled by three set of alleles. Okay, what are the three alleles? IA, IB, and small I. Now the alleles IA and IB are equally dominant and are said to be codominant. So both of the alleles are equally dominant. Both are expressed in the heterozygous condition. Now both these alleles do not interfere with the expression of each other. That's why both are being expressed. In case of dominance, what happens? One of the allele is masking the effect of the other. But here, both these alleles in codominance, they do not interfere with the expression of the other alleles. Hence, AB blood group is an example of codominance. In the person having AB blood group, the plasma membrane of the RBCs will consist of both the sugars produced by IA and IB. So you can see there are differences in the RBCs of the person having different blood group, basically on the sugars which are present on the plasma membrane. So you can see this is table. This is showing you the genetic basis of blood groups in the human population. Now you can see the individual, the diploid organism, it acquires one allele from one parent, another allele from the other parent. So this is allele from one parent, allele from the other parent. This is the genotype that is shown to you. And in the last column, the phenotype is shown to you, or the blood type of the offering is being shown to you. Now the parent is having IA allele and the other parent is having IA. So the offerings will be having IA and IA genotype, and it will be having A blood group. Then one of the parent is having IA, another parent is having IB. So in this case, both the alleles are inherited. by the offerings, and the blood group will be AB here. So here you can see codominance. Then one of the parent is having IA and small I. So genotype will be IA and small I in the offerings, and this will be having A blood group because IA is dominant over small I. Then you can see again another parent. Okay, parent 1 is having IB, parent 2 is having IA. The offerings will be having IA and IB. Blood group will be AB. Clear? Then if the parent is having IB, another parent is having IB, the genotype of the offerings will be IB, IB. Okay, the blood group will be B. Okay. Then next one is IB and small I. Genotype will be IB and small I. Okay. The blood group will be B because IB is dominant over small I. And if both of the parents contain small I, then the genotype is small I. Okay. And the blood group will be O. So for small I, these children's or these offerings, they should inherit small I from both the parents. So here you can see there are 6 genotypes and there are 4 phenotypes. What are the 6 genotypes here? 6 genotypes are IA, IB, IA small I. Clear? Then IB and IB small I and small I. So these are the 6 genotypes, but there are only 4 phenotypes. What are the 4 phenotypes? A blood group, AB blood group, B blood group, and O blood group. These are the 4 phenotypes. Clear? Now let's talk about incomplete dominance. Now it is a phenomena in which one dominant allele shows incomplete dominance over other member of the allelic pair of a character. Or you can say in other words, neither of the allele is dominant over the other. Okay? Or one allele shows incomplete dominance over the other. This is another way of writing incomplete dominance. So, due to this, there is a blending of the characters. So, this leads to blending of the traits. And you get an intermediate phenotype, which is different from both the parents. For example, flower color is snapdragon, which is Antirrhinum majus. Now, a monohybrid cross between the plant having red flowers and plant having white flowers. This will result in the pink flowers in the F1 generation. So in F1 generation, you are getting pink flowers instead of red flowers. Okay, or white flowers. You should get red flower actually because red color is dominant over white color. So actually you should get red flowers here. But you are getting pink flowers. So this is due to incomplete dominance. Okay. So here you can see this is the cross between red flower plant and a white flower plant showing incomplete dominance. Okay. So let's have a look at this image. So you can see the parents, one of the parents is having red flower, another parent is having white flower. The parent having red flower, this is the genotype, capital R, capital R. The parent having white flowers, it has the genotype, small r, small r. Okay, clear? Both are homozygous. One is homozygous dominant, another one is homozygous recessive. Now the gametes produced by them, the red color flower, okay, this parent will produce only one kind of gamete. All the gametes will contain capital R allele. Another parent having smaller, this will produce gametes having small r. Okay, now you get the F1 generation after fertilization of the gametes. All the F1 generation will have pink flowers and they have the genotype capital R, small r. So in the heterozygous condition, F1 hybrid, this is heterozygous, you will get the pink phenotype here, pink flowers. Clear? This is because neither of the allele is dominant over the other. Now, after selfing of the F1 generation, you get the F2 generation. Now each of the parent will produce two kind of gametes. Half of the gamete will contain capital R, half of the gamete will contain small r. You can make the Punnett square now. Okay, at one side you will write male gamete, another side you can write the female gamete. Then you can make all the possible combinations here. Now you have to remember one thing here. The phenotypic ratio is 1:2:1. So usually in a monohybrid cross, okay, which Mendel did, phenotypic ratio is 3:1. So this 3:1 ratio you get when the genes are showing, when the genes are following law of dominance. Okay, when incomplete dominance is, you can say, is observed, then the phenotypic ratio changes and it becomes 1:2:1 instead of 3:1. And genotypic ratio is 1:2:1. So here in phenotypic ratio, 1 is red, 2 is pink, 1 is white. Okay, clear? So in incomplete dominance, the phenotypic ratio and genotypic ratio both are 1:2:1. So you have to remember this. The next question, what is point mutation? Give one example of the point mutation. Clear? So what will be the answer for this question? So let's have a look at the answer. So point mutation, this is a change in the single base pair of the DNA. Okay, or change in the single base of the DNA. Okay, so bases, they are complementary to each other in the DNA because DNA is double helix. It consists of two strands. So we can write change in single base pair or change in single base of the DNA. If there is a change in the single base, obviously the complementary base will also change. That's why we write single base pair also. So both are correct. Clear? Now change is caused by substitution, deletion, or insertion of a single nitrogenous base. Clear? So basically it is substitution only. A single base pair is substituted by some another base. Okay. Example of point mutation is sickle cell anemia. Okay, what happens in sickle cell anemia? Here there is a change, there is a mutation in the single base pair at the 6th position of the beta globin chain. Okay, so hemoglobin, it is made up of globin chains, alpha globin and beta globin. So there is a mutation at the 6th position of the beta globin chain. Okay, this causes sickle cell anemia, and because of this single base pair mutation, glutamic acid is replaced with valine at the 6th position of the beta globin chain. So you can see in this image, this normal hemoglobin, this have GAG, but in sickle cell condition, it becomes GTG, means A is replaced by T. Okay, then DNA is double helix, so there is GAG in the normal hemoglobin. Corresponding, it will complementary bases will be CTC. Here in the case of sickle cell, it was GTG, complementary sequences will be CAC. So this is one codon they are showing. Okay, at the 6th position of the beta globin chain. Now mRNA is produced from DNA, so mRNA is also changed. It should be GAG, normal mRNA, but it becomes GUG now. Okay, so GAG codes for glutamic acid, GUG codes for valine. So glutamic acid is replaced by valine at the 6th position of the beta globin chain. Yeah, so this is due to single base pair mutation. Clear? And now the hemoglobin polymerizes because of this change, because of this amino acid substitution, and the RBCs become sickle shaped now. So sickle cell anemia is a classical example of point mutation. It is an autosomal recessive disorder. Okay, this is clear. The next question, who had proposed the chromosomal theory of the inheritance? This is a very straightforward question. They are just asking who has proposed the chromosomal theory of inheritance. So what will be the answer for this question? So let's have a look at the answer now. The chromosomal theory of inheritance was proposed by Sutton and Bovary. They proposed the chromosomal theory of inheritance in 1902. Clear? Now, they linked the inheritance of trait to the chromosomes. So, what Sutton and Bovary said? They said that the genes are present on the chromosomes, and genes and the chromosome, both of them, they behave in the same manner. What does this mean? They behave in the same manner. So, if the genes are segregating, then if the chromosomes are segregating in a similar fashion, the genes will also segregate. Okay, they will follow the same segregation pattern because genes are located on the chromosomes. Okay, genes are linearly arranged on the chromosomes. The chromosomes are segregating in a certain pattern, then the genes will also segregate. So their behavioral parallel to each other. Both of them behave in a similar manner. And what did Bovary and Sutton said in chromosome theory of inheritance? Okay, so they explained Mendel's law using chromosome movement. So they studied the chromosomes movement. So now at that time there were very advanced microscopes. You can study the cell division under this microscope. So you can see the movement of chromosome now. You can stay in the chromosome and you can see the movement of the chromosome. So they basically, you can say, explain Mendel's law of inheritance by chromosome movement. So they said chromosomes are the physical structure. Okay, you can see them. Okay, and genes are present on the chromosomes. So chromosomes are the physical structure by which we can explain Mendel's law of inheritance. Clear? So this was chromosomal theory of inheritance given by Sutton and Bovary. You should remember the year also, in 1902. Okay, in short, they linked the inheritance of traits which are controlled by the genes to the chromosomes. Okay. The next question, you have to mention two autosomal genetic disorders and their symptoms. So they are just asking straightforward questions here. So these genetic disorders, they can be linked to autosomes or they can be linked to sex chromosomes as well. You have to just mention two autosomal genetic disorders with their symptoms. So for more information, you can read the NCERT section 4.8.2. Okay. So let's have a look at the answer for this question. So autosomal genetic disorders, they are asking about autosomal genetic disorders. So these are disorders in the autosomes. So like humans, they have autosomes and they have sex chromosomes also. In humans, you have 46 chromosomes. Okay, means 23 pair of chromosomes. Out of this 23 pair of chromosomes, you have 22 pairs of autosomes and one pair of the sex chromosome. Okay, clear? Now, if there is some disorders in the autosomes, it is known as autosomal genetic disorder. So conditions caused by mutation in genes located on the autosomes. This is known as autosomal genetic disorders. Okay, clear? So these genetic disorders are also known as Mendelian disorders. So genetic disorders are of two types, Mendelian disorders and chromosomal disorders. So these are Mendelian disorders which are caused due to mutation in a single gene. Okay. Now, two autosomal genetic disorders are as follows. One is thalassemia. This is autosomal recessive disorder. Okay, so this is autosomal recessive blood disorder. So you should know the inheritance pattern of autosomal recessive blood disorders. That they skip generations and both the parents should be carriers. Both males and the females, they are equally affected. Okay, so you should remember this. You should remember the difference between autosomal dominance inheritance and autosomal recessive inheritance. So what they will give you a pedigree chart. Okay, they will give you a family tree, and then they can ask you, tell the inheritance pattern, whether it is autosomal dominant, autosomal recessive. You get this type of questions in your NEET exam as well. Okay, they can ask you whether it is linked, sex linked, or whether it is autosome linked. Okay. Now in thalassemia, what happens? Less amount of hemoglobin is being produced because reduced amount of globin chains are being produced. So thalassemia is caused due to mutations or deletions in the genes. And it is of two types, alpha thalassemia and beta thalassemia. In alpha thalassemia, what happens? There is a reduction in the production of alpha globin genes. In beta thalassemia, there is a reduction in the production of beta globin genes. Okay. So that's why it is the quantitative disorder. Here, less amount of globin chains are being produced. And due to this, the person suffers with anemia. Clear? Some other abnormalities can also occur. Clear? Another one is phenylketonuria. Sickle cell anemia is also autosomal recessive disorder. It is due to point mutation. Phenylketonuria, this is also autosomal recessive disorder. In phenylketonuria, what happens? It is a metabolic disorder. Clear? And it is a very good example of pleiotropy also. Remember this, pleiotropy is what? What is pleiotropy? When one gene affects multiple traits. Clear? Phenylketonuria, what happens? Here a gene, there is a mutation in a gene, and this gene codes for an enzyme, phenylalanine hydroxylase. So due to mutation, this enzyme is not being produced, or this enzyme is non-functional. Clear? And this enzyme, what is the function of this enzyme? This converts phenylalanine into tyrosine. Now, due to lack of this enzyme, phenylalanine is not converted into tyrosine, and phenylalanine accumulates in the body. Further, it gets converted into phenyl pyruvic acid and other harmful compounds. And these compounds, they affect the brain. So it causes mental retardation as well. It can cause brain disorders. Along with it, it can also cause reduced, you can say, pigmentation of skin and hairs. So here you can see a single gene is affecting multiple traits. That's why it is example of pleiotropy. Clear? Here there is accumulation of phenyl pyruvic acid and other derivatives in the brain because the enzyme phenylalanine hydroxylase is being affected. And this results in poor skin pigmentation, okay, mental retardation, they are reduced hair pigmentation as well. Okay. And another one we have already discussed, sickle cell anemia. Here there is a mutation at the 6th position of the beta globin chain. Okay, so you should remember all the genetic disorders apart from, okay, so genetic disorders, as I have told you, there are two types, Mendelian disorders and chromosomal disorders. So chromosomal disorder, they are due to changes in the chromosome number or in the change in the chromosome structure. Okay, so within chromosomal disorders, you should study Turner syndrome, Klinefelter syndrome, and Down syndrome. Okay, these are also genetic disorders, but they are characterized into chromosomal disorders. Clear? In Down syndrome, what happens? It is a trisomy of the 21st chromosome. So again, here autosomes are involved in Down syndrome. But in Turner syndrome and Klinefelter syndrome, the sex chromosomes, it is about sex chromosome. In Turner syndrome, what happens? There is a lack of one X chromosome in the females. In Klinefelter syndrome, what happens? There is one additional X chromosome in the males. Clear? So you should remember all of these genetic disorders. Okay. So with this, we end this session. I hope this session was quite knowledgeable for you. We have completed the exercise for this chapter. We'll meet you in the next session. Thank you very much for listening.